Tuberculosis is a disease caused by Mycobacterium tuberculosis. This disease is spread through close quarters contact like coughing, and symptoms include coughing up blood, bad cough for over 3 weeks, weakness or fatigue, and no appetite, chills, or weight loss. Mycobacterium tuberculosis is normally treated with antibiotics, and it usually destroys the cells. But over several generations, it starts to become resistant to the drugs, and this results in mutations or other transformations in the bacterium. And the mutated Mycobacterium tuberculosis is resistant to the antibiotics. This makes us look for different approaches in treating the bacteria. This is Mycobacterium tuberculosis. The cell wall is represented by the outer rubber, and this is represents the peptidoglycan layer and the lipid bilayer. And on the inside, we have candies and air, which is going to represent the cytoplasm and the organelles. As we break the cell wall, that's very tough to do. The cell becomes unstable, and the wall breaks, exposing the cytoplasm and the organelles. This allows for the cell to be either totally annihilated or just eaten by a white blood cell or other things. And it is very delicious.
This is a picture showing the peptidoglycan layer of the bacteria cell. This is made up of a polymer of NAG and NAM. The NAM is shown with the blue hexagons and the NAG is the purple. Dipeptide chains, which are shown down in the picture by those yellow circles, combine the two NAM molecules. If we could find a way that we could break the dipeptide chains, we could greatly weaken the bacteria cell to treat the M. tuberculosis that is multi-drug resistant. To get a better understanding about where and how we can cut these dipeptide chains, we made a 3D model of this peptidoglycan layer.
This is a model of the peptidoglycan layer of the bacteria cell, also known as the cell wall. It's a branched polymer. Note here where it alternates between NAG and NAM. On each NAM, there is a dipeptide chains represented by the white ropes that connects each NAM. The NAG do not have this. These dipeptide bonds can be made up of molecules like D-alanine, D-glutamate, or other molecules. Notice that they give support to the cell as seen by them holding up a pen. If this dipeptide chain is broken, the cell will lose its structural support and the cell will be weaker. Once the cell is weakened, it will make it easier for molecules of the immune system to attack the cell and have it die. This is DDL, dealanine-dealanine ligase. DDL is a dimer, meaning that it's the same on this side that it is on the other side. Because of this, what's happening on this side is also going to happen on this side as well. To simplify the mechanism for explanation, I'm just going to explain one of the sides, but just note that the same thing is happening on both sides. Before I start to talk about the mechanism, I'd like to point out that DDL requires 2 dealanines and 1 ATP to work. Notice that it has 2 active sites, both for dealanine, and 1 active site, which is for ATP. Before the reaction will start, notice that ATP in the current conformation cannot bind to DDL. To allow for the binding of ATP, there has to be a conformational change to occur. This conformational change happens after the first dealanine will bind to DDL. This conformation is a change in conformation at the omega loop. After there is a change in conformation, it allows for the binding of a second dealanine and for the binding of ATP. Notice that the active site on this dealanine is represented by a triangle in the model, and this one also has a triangle as well. However, in our model of DDL, the first active site is represented by a triangle, where the second active site is represented by a kind of triangle, but it's also square at the bottom. Notice when a second dealanine comes to bind in with DDL, D-alanine can kind of fit in the active site, but DDL is going to have a lesser affinity for this diala. This is going to come to be important later on when we talk about inhibitors for DDL. Since the diala is not going to bind to DDL very well here, this is a good site for an inhibitor to attack. This is D-cycloserine, which is going to serve as an inhibitor for the reaction of D-alanine-D-alanine ligase. In the presence of DCS, the first diala is still going to bind to DDL. When the first diala binds, there's still going to be that conformational change at the omega loop. However, this is going to expose the second active site for DDL. Since I said before that this diala has less of an affinity here for DDL, this lets the DCS bind here. Notice when DCS is bound here, ATP cannot bind to D-alanine-D-alanine ligase. Because ATP and two dialas cannot bind to D-alanine-D-alanine ligase, this is not going to form a diala-diala dipeptide, which can be used in the cell wall. Because this dipeptide cannot form, the cell wall is going to be weaker, and that would make the cell susceptible to an attack by the cells of the immune system, which would later kill the cell. Another way that DCS can inhibit D-alanine-D-alanine ligase is by binding at the first site that the first diala was supposed to bind. Notice that we made our model to make DCS kind of fit in the first active site, but not as well as diala can fit in the first active site. Notice that in the model, DCS fits so much better in the second active site. In fact, DCS even fits better than diala. This was done to represent the fact that even though DCS can still inhibit the reaction at the first site, diala is much better at binding to DDL at this site. Because of this, it's much better at inhibiting the reaction at the second site.